Mitosis. Okay, this is chapter 16. With cell division. The adult human body contains between 10 to 15 trillion cells. Cell division is the reproduction of these cells. The eukaryotic cell follows one of two different sorting processes. Okay, we start off with mitosis, which means that there are going to be two daughter cells that receive the same amount of genetic material as the mother cell. And then we have meiosis, which is needed for sexual reproduction so that cells have two sets of chromosomes, such as two parents, and produce daughter cells with a single set of those chromosomes. Eukaryotic cell cycle. Okay, we have this omnis cellula e cellula, which is every cell originates from another cell, which we know is part of cell theory. All organisms produced by a series of repeated rounds of cell growth and division, extending back to the beginnings of life nearly 4 billion years ago. The cell cycle itself is a highly regulated series of events that leads to the cell division. These eukaryotic chromosomes. We have what's called cytogenics, which is the field of genetics involving microscopic examinations of the chromosomes. When cells get ready to divide, the chromosomes become compact enough to be seen with a light microscope. With microscopic examination of these chromosomes, white blood cells stimulated to divide, the cells caused to expand, and they treated with a fixative. Okay, this stain is a chemical dye that stains chromosomes, which leads to what's called G bands. This gives us a karyotype, which reveals the number, size, and form of chromosomes in an actively dividing cell. In a karyotype, two copies of each chromosome are still joined together, and these are called sister chromatids. This here's the overall process of how to make a karyotype, but what I want you to focus on is this diagram, this picture right here. Okay, this is showing our 23 chromosomes, okay, and how they are paired together. Sets of chromosomes. Humans have 23 pairs of the chromosomes, or 46 total chromosomes. Okay, they're numbered according to size. Autosomes are the first 22 pairs in humans, and the sex chromosome, we only have one pair, and it's either XX for female or XX for male. Ploidity means we have a diploid or 2N, so humans have 23 pairs of chromosomes, or a haploid or N, which is a gamete, which is either a sperm or an egg of a sex cell, have one member of each pair of chromosomes, or 23 total chromosomes. What are homologs? In diploid species, members of a pair of chromosomes are called homologs. They are homologous chromosomes evolutionarily derived from the same chromosome. We then have autosomes, each homolog nearly identical in size and genetic composition. For example, both carry genes for eye color, but one may have brown and the other blue. In most cases, homologs differ by less than 1%. The sex chromosomes means X and Y are very different from each other in size and composition. Now we move on to the cell cycle. We have growth phase. So G1 of the cell cycle is growth phase, and it's a part of interphase. Signaling molecules can cause cells to accumulate molecular changes during G1 that promotes progression through the cell. Here we have synthesis phase. The second part of interphase is known as S phase of the cell. This is when DNA replicates. Chromosomes replicate here to form a pair of sister chromatids. And after replication, a cell has twice as many chromatids as the number of chromosomes in G1. In growth phase 2, or G2, the third part of the cell cycle, this is just a checkpoint to make sure that the DNA was correct, that replicated in the S phase. Here, the cell itself prepares for division. Cell synthesizes proteins needed for the chromosome sorting and cell division, and some growth may occur here. Then we have stage 4 mitosis. This is the last part of the cell cycle, and it has 4 phases during which for which the cell divides into 2 daughter cells. Then we have cytokinesis. This follows mitosis to divide the cytoplasm into 2 daughter cells. Lastly, we have cancer, but cancer is not a phase. This happens at this stage if the cell is mutated and continues to divide. This means something went wrong in the DNA. Cell cycle length. Several minutes in quickly growing embryos. It takes several months in slow growing adult cells. For fast dividing mammalian cells in adults, which are skin cells, it takes them only 10 to 24 hours. For a cell that divides in 24 hours, okay, 11 hours is devoted to G1, 8 hours is devoted to S, 4 hours is devoted to G2, and 1 hour is devoted to mitosis. Decision to divide. Okay, this depends on many external factors, such as environmental conditions and signaling molecules. It also depends on internal factors, such as cell cycle control molecules and different checkpoints. Those checkpoints, we have what are called checkpoint proteins. These are cyclins or cyclin-dependent kinases or CDKs, and they're responsible for advancing a cell through the phases of the cell cycle. The amount of cyclins varies through cycle, and these CDKs are kinases controlling the cell cycle, and they must bind a cyclin to be.
The process of cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis, M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and 2 daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
Diagram of the cell cycle for you to study as well, going step by step. Diagram of the checkpoints that the video went over. Okay, so then we have the cell cycles checkpoints. Okay, these are critical regulatory points. There are 3 checkpoints in the eukaryotic cell cycle. You have the G1 checkpoint, which is the restriction point. Oh, this is a mistake on the slide. This should read G2 checkpoint. Okay, and then we have the metaphase checkpoint. The checkpoint proteins act as sensors to determine if a cell is in proper condition to divide. The cell cycle is delayed until problems are fixed, or it is prevented altogether. There are 3 main checkpoints, as we mentioned in the last slide. We have the G1 checkpoint, which is a restriction point. This is where proteins determine if conditions are favorable for cell division. It actually senses the DNA damage. We have the G2 checkpoint, which is a check for DNA damage, and it is there to ensure that all of the DNA has been replicated. It monitors the levels of proteins needed to advance this through mitosis. And then we have the metaphase checkpoint. Here it monitors the integrity of the spindle apparatus. It prevents cells from incorrectly sorting their chromosomes during the division. Loss of checkpoint genes due to mutation increases the likelihood of an undesirable genetic change, more mutations, and cancer. Going on to mitosis. This is the basic phases of a cell's life is interphase, mitosis, which contains prophase, metaphase, anaphase, telophase, then cytokinesis. In preparation for cell division, the DNA is replicated. The sister chromatids, okay, are two identical copies with associated proteins. Chromatids are tightly associated at a centromere. The sister chromatids are held together by cohesion. This serves as a detachment site for uh, kinetochore used in sorting chromosomes. So here we have a diagram for the replication of those chromosomes. Okay, so it would be good of you to study the steps 1 and 2. Start off with interphase. This is the longest stage of a cell's life. The time is spent here when it's not dividing. This phase produces all materials that are required for growth, and it is mostly there for preparation for division. Next, we have prophase. Okay, what we're showing here is actually early prophase. The chromosomes get small. The centrioles move to the poles of the nucleus. The spindle fibers then develop to late prophase. Here, this happens when the nuclear envelope disintegrates or disappears. The spindle fibers begin to move chromosomes toward the center of the cell onto metaphase. This means that the nuclear membrane is now completely gone, and the duplicated chromosomes line up along the cell's equator. Okay, so here we see the equator for this cell. Now it's important to note that the cell's equator can be either pictured horizontal like this or vertical. Okay, so here we see metaphase along up the equator, and here we can slightly see those spindle apparatus pulling and pushing the chromosomes. Anaphase. This is where the diploid set of daughter chromosomes separate. They are pushed and pulled toward the opposite poles of the cell by those spindle fibers. Okay, here we can see an early anaphase where they're being pushed and pulled apart to the poles of the cell. Telophase. The nuclear membrane and the nucleoli, or the nucleus, start to reform. We then have a cell plate that begins to form in plants. And then we have what's called a cleavage furrow that is starting to form in animals. Cytokinesis, the cytoplasm, organelles, and nuclear material are evenly split, and then two new daughter cells are formed.
Cytokinesis is the phase of the cell cycle during which the cytoplasm actually divides. It follows nuclear division and involves the splitting of the cell into two cells. In animal cells, cytokinesis is achieved by a constricting belt of actin filaments interacting with myosin. As the filaments slide past one another, the diameter of the belt decreases, pinching the cell and creating a cleavage furrow around the cell's circumference. Constriction proceeds and the furrow deepens until it eventually slices all the way into the center of the cell. At this point, the cell divides in two. In plant cells, membrane components are assembled in the center of the cell to form the phragmoplast. These components are vesicles derived from the Golgi apparatus. The vesicles travel along microtubules, and their content forms the cell plate. The cell plate continues to grow outward until it reaches the cell membrane along the perimeter of the cell. Cellulose contained in the vesicles is then laid down between the new membranes, creating two new cell walls. Pectin is also deposited in the space between the daughter cells.
Of mitosis and cytokinesis. Okay, we have two new daughter cells with the same number of chromosomes as the mother cell. Okay, barring mutations, the two daughter cells are genetically identical to each other and to the mother cell from which they were divided or derived. Diagram shows anaphase. A, B, C, or D? The answer is C.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads-on-a-string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the 2 identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Next with this lecture, we have meiosis. The easiest way to differentiate the difference between mitosis and meiosis is just the simple phrase, meiosis makes me. That way you can remember that they're the sex cells, they deal with the gametes, and that they divide more than mitosis cells. So here we have meiosis. Meiosis is the process by which haploid cells are produced from a cell that was originally a diploid. So an example of this is the diploid human cell has 46 chromosomes, but after meiosis, the haploid egg or sperm has 23. Okay, so we get 23 from our mom, 23 from our dad, and that makes 46, which is us. Okay, that means it goes through 2 rounds of division. Okay, and it's called meiosis 1 and meiosis 2. So what's some of the differences between mitosis and meiosis? Like mitosis, meiosis begins after a cell has progressed through the G1, S1, and G2 phases out of the cell cycle. But two of the key main differences is with meiosis, you have a homologous pair form a bivalent or tetrad, and then there's also crossing over that happens. So what that bivalent or tetrad is, it's homologous pairs of sister chromatids that associate with each other lying side by side to form a bivalent or a tetrad. This process is called synapsis. Okay, and we have synaptometal complex, which is a protein structure that connects the homologous chromosomes together. However, it's not actually required for chromosome. However, it's a little bit more important with this lecture. It's the physical exchange between chromosome pieces of the crossing bivalent. I'll show you a picture of what that here is in a moment. So this may increase the genetic variation of species. Okay, we have what's called a chiasm, which are arms of the chromosomes that tend to separate but remain or adhere at a crossover site. A number of crossovers are carefully regulated. Here's the diagram. So we first have the homologous chromosomes that condense. As they're side by side, this is that synaptonomial complex that forms. Okay, the chromosomes become bivalent. Okay, and we see up here this little crossing over. It's almost like they switch places in the chromosomes. Okay, and so they have crossed over to form something different between you and your brother or your sister.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
Although mitosis and meiosis have much in common, meiosis has 3 unique features: synapsis, homologous recombination, and reduction division. Following chromosome replication, homologous chromosomes, or homologs, pair all along their length. The process of forming these complexes of homologs is called synapsis. The second unique feature of meiosis is that genetic exchange occurs between the homologous chromosomes while they are physically joined. The exchange process that occurs between paired chromosomes is called crossing over. The third unique feature of meiosis is a reduction division. At the end of meiosis, each cell contains only half the original complement of chromosomes. Because of the crossing over that occurs during the first division, the sister chromatids in meiosis II are not always identical. Omitting a chromosome duplication before meiosis II produces haploid gametes.
Meiosis I, it is also interphase. Here we have the chromosomes replicate. The chromatin contains 2, which is prophase I. We have the pairing of homologous chromosomes occur. Each chromosome consists of 2 chromatids, and the nuclear membrane starts to break down. The spindles are also forming. A continuation of prophase I. This is where crossing over produces the exchange of that genetic material, which is why we're different from our brothers and our sisters. Crossing over itself is the chromosomal segments are exchanged between a pair of homologous chromosomes as shown in the videos. 1. The chromosomes centromeres attach to the spindle fibers. The homologous chromosomes line up at the equator. And at this point, we should be noticing this is very, very similar to mitosis so far.
During metaphase I of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
Here the homologous chromosomes separate and move to opposite poles of the cell. Is 1. Here we have spindle fibers break down. Chromosomes uncoil and they form 2 nuclei, and the cell divides.
Meiosis follows interphase after the chromosomes of a diploid cell have replicated. DNA replication occurs during S phase. After the 2 successive cell divisions of meiosis, a single cell will produce 4 cells, each having the haploid number of chromosomes. Prophase I of meiosis I begins with the condensation of chromosomes and the vesiculation of the nuclear membrane. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, synaptonemal complexes form, and the homologous chromosomes are seen to align next to each other to form a structure called a bivalent. A bivalent is composed of 4 sister chromatids. Crossing will occasionally occur between homologous chromatids within a bivalent. During prometaphase 1, the spindle apparatus continues to form and bivalents become attached to kinetochore microtubules. One pair of sister chromatids is connected via kinetochore microtubules to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The complete assembly of the spindle apparatus occurs during prometaphase 1. The chromosomes align at the metaphase plate during metaphase 1. It is important to notice that bivalents are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase 1 involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the kinetochore microtubules shorten. When the conjoined sister chromatids reach the poles of the cell, they detach from the spindle apparatus, and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis ultimately results in 2 cells, each having the haploid chromosome number, with each chromosome consisting of a pair of sister chromatids. This completes meiosis 1, and after a period of interkinesis, the 2 daughter cells start the second cell division cycle called meiosis 2.
Meiosis 1, and now we're on to prophase 2. Okay, a second set of phases begins as the spindle apparatus forms and the chromosomes once again condense. On to metaphase 2, a haploid number of chromosomes line up at the equator. 2. Here the sister chromatids are pulled apart at the centromere by the spindle fibers and moved toward the opposite poles of the cell. Telophase 2, where the chromosomes reach the poles and the nuclear membrane and the nuclei reform. Cytokinesis. This results in the 4 haploid cells, each with an n number of chromosomes.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber, and in telophase 2, along with cytokinesis, we see the formation of four genetically different haploid cells.
A chromosomal mutation is a substantial change in the structure of a chromosome or chromosomes. One way that this can occur is by a deletion or a duplication within one chromosome. In the chromosome, shown here, the longer arm is labeled Q and the shorter arm is labeled P. Regions are designated with numbers. If a region is deleted, such as a large portion of the Q2 region shown here, then a deficiency occurs. There is now less genetic material in the mutated chromosome. A duplication occurs when an extra copy of a chromosomal region exists, such as the extra copy of regions Q2 and Q3 shown here. Both duplications and deletions can be created by abnormal crossing over during meiosis. An inversion occurs when a chromosome is broken in two places and reconnected so that a region is flipped from the normal order, such as the Q2 and Q3 regions shown here. Note that an inversion does not change the total amount of genetic material in the chromosome. Translocations involve two chromosomes. In a simple translocation, a single piece of one chromosome breaks off and is attached to another chromosome. In a reciprocal translocation, two chromosomes exchange pieces. Note that translocations do not change the total amount of genetic material. However, both inversions and translocations can lead to overall deficiencies and duplications of genetic material after meiosis.
An inversion is a reversal in the order of a segment of a chromosome. Inversions do not alter gene expression unless the breakpoints on the ends of the inversion occur within a gene. When a segment of a chromosome is inverted, it can pair in meiosis only by forming an internal loop. Although crossing over occurs in the four-strand stage during meiosis, only two strands are shown for clarity. Crossing over that occurs within the inverted segment during meiosis can result in some genes being lost on one chromosome, while others are duplicated on another chromosome.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase II, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase II, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase II. During anaphase II, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase II, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.